there's no point.
Maybe there's just like random kind strangers out there. Oh, no story, sure. I punched a good today, so.
It would have to be people.
So if you don't work, do you like a sugar baby too? Or milk? Okay. Okay. So is he like, is he dog status?
I would say no. He's a dad.
Okay.
I wouldn't say no. Yeah. Just like dad. Not milk.
What's his name? He's so soft.
Here, like, take this off while you're talking. All right, everyone, find a seat, find a seat. Everyone, come forward! Come forward! Hello, hi. Good to see you. Wave to the camera.
I don't want to give it.
All right, cool. Hey everyone, I'm Eleanor, I'm head of DeSci here at DeSci. This is the DeSci HQ. Thanks very much for coming and super excited to be hosting the DeSciNYC. We're going to be hosting an exciting talk on cellular metabolism, so that'll be coming up very shortly. A little bit about SAVE, so we're at layer 1, fastest EVM parallelized blockchain. We're also dedicated to decentralized science, so as well as other applications in crypto, but also a focus on the application of crypto in the sciences, AI, frontier technology, robotics, etc. We also have a fund, so 65 million in capitals, called Sapien, dedicated towards building projects on SAVE. Yeah, so I mean, that is pretty much everything. I hope you enjoy the talk, and Mike, I'll hand over to you for a couple of notes on DeSciNYC.
Guys, let's give it up for Say. They gave us this beautiful office, and we're so appreciative for them. And thank you to Eleanor and the entire Say team. And look at this, we have a koi pond over here. How cool is that? Okay, let's go to the next slide. How do I do it? Who wants a sleep mask? Okay, while we're waiting for the next slide, we'll do a sleep mask. A sleep mask. A sleep mask. Oops, sorry. A sleep mask. A sleep mask. All right, in the far back. Sorry. Sorry. Oh, is this? Oh, where's this slide? Okay. Okay. Okay, so guys, welcome to DeSciNYC. We host these events like once a month, and you know we have one coming up next time on quantum computing, which is going to be very, very cool. And I'm going to give it to Avinash right here. Yes, he's going to talk about a new type of clinical trial that we're going to be working on.
Thank you, Michael. Hello, everyone. You probably are aware that the microbiome can impact all sorts of aspects of health, but it's still in the very early stages of figuring out how, and most of that focuses on the negatives of how certain strains of bad bacteria can cause problems. But there was recently a lab in MIT that decided to go and look at a bunch of elite athletes and look at their microbiomes and try to figure out which species might be common amongst a lot of elite performers that could potentially lead towards part of their actual success. They were able to isolate a bunch of different strains. Right now, they ran some animal trials, some human trials, they're on the market. I have no actual association with this company in terms of there's no financial link. I just like them, and we're partnering together with Michael to basically take 200 of you, and whoever decides to join, give you free access to 2 different types of these probiotics and figure out at a larger scale, can that impact sleep, digestion, exercise performance? These are all things that have been validated fairly well within the rodent studies and the early human trials, and we're not ones to take anything with small amounts of data and say it absolutely works. So instead we want to get our own larger amounts of data, run our own trials in a decentralized way, and figure out, can we take this and measure its impact on human performance in terms of sleep and physical activity? So if you're interested in getting free access to this fairly expensive probiotic and are willing to do the work to try and test it in yourself and measure the outcomes, come and talk to me or Michael, or there's a QR code somewhere where you can sign up.
Yes.
Excellent. Find us afterwards.
But what's cool about this is DeSciNYC for so long has been just about looking at content and learning about content. But this is one of the first times that a group like this is going to actually be creating new types of science and contributing to the canon of knowledge. So this is going to be very, very cool. You've helped design this study as well. Yeah? And this I don't know if you know, but he's like an influencer on science, and you should give a talk at some point. I think at some point you will. And maybe you could even talk about the results and how doing it in a decentralized way is possible now.
Yeah, that'd be fun.
Cool. Do we have the link?
Yeah.
Okay. I just want to get the sign up link here.
In case I downplayed it, a lot of people have been reporting really cool results from it, but it's always hard to tell from placebo, so that's why we want to get good data.
Okay, and then we're getting the link up here, and then people that are interested in it. So this is part of DeSciNYC, and it's going to be one of the first trials that's ever been done in a new way. So I'm very excited about this. I don't think that people at crypto and tokens and democratization of science has been done in this way. So I'm very excited. Okay, so without further ado, we are going to bring Timothy on stage. Okay, so let's give a round of applause for Timothy. Timothy is a postdoc at the Rockefeller University studying cellular metabolism. So let's give it up.
Thank you guys very much. I'm super excited to be here and present the work that I've been working on in my postdoc, and I'm starting my own laboratory at Mount Sinai in January. So I'm going to be talking a little bit about the stuff that keep me up at night, and I'm excited to have a different audience to talk to than a normal academic audience. And I think too, one of the cool things about this, about DeSci and about decentralizing science is finding new revenue streams to fund science, to fund basic science that companies necessarily aren't going to be funding. It's very important research for us to be doing. And I hope I can convince you of some of that through my talk and kind of the stuff that I've been working on. Okay, so I will be, so the focus of my talk is really cellular metabolism. And what is metabolism? Metabolism is the sum of all chemical reactions within a biological system. And this is a map showing kind of the central metabolic pathways in mammalian cells. And I know it looks quite complicated, but this is actually a gross oversimplification of metabolism. So we know that there's over 220,000 reported human metabolites in humans. And just in human serum, for example, there's over 4,500 serum metabolites. And so one of the questions that I and so many people for centuries and millennia have been interested in is how is metabolism regulated and controlled, and how do humans and living organisms maintain metabolism to sustain life? And so Aristotle started thinking about this in the 4th century BC, and he made the observation that the food that we eat must be what provides the energy and the building blocks that we need to grow and sustain life in humans. And a couple centuries later, Sanctorius of Padua actually expanded upon this and took very detailed measurements of his consumption and excretion over 30 years and made the observation that for every 8 pounds of food he ate, only 3 pounds of waste was produced. So therefore he said this must be going somewhere. And that principle has really been expanded at the cellular level. So in the 19th century, Ernest Overton studied how metabolites get in and out of cells and how they are transported into the cell and how that enables them to be used for different cellular metabolic pathways and processes. And so really, the key player of cellular metabolism is transporters. And transporters are proteins that sit on the cell membrane that bring nutrients in and out of the cells. And this is a really fundamental process for cellular biology and organismal biology in general. And the fact that this is so important is highlighted by the fact that over 10% of the human genome, all genes that humans encode, they encode for proteins that have transport function. So it's a very key function of cellular homeostasis, but also organismal homeostasis in life. And one of the largest families of these cellular carriers is known as the cellular carrier superfamily, the SLCs. And there's over 450 of these different transporters, which range in diversity of where they're located within the cell, what they transport, what affinity they have for their substrates. And throughout evolution, these different transporters have evolved to enable organisms and cells to tightly regulate their metabolism. So for example, if we take glucose as an example, which is a very abundant metabolite, there's at least 4 different glucose transporters. And they change in where they are expressed in the human body. So for example, in the brain, you have this transporter called GLUT3, and that has very, very high affinity, meaning it really, really wants to transport glucose. And that makes sense because the brain has high energetic demands. In other tissues, such as in pancreatic beta cells, the affinity of the transporter GLUT2 has very low affinity for glucose, and that allows it to act as a sensor so that your pancreas can understand whether or not you've recently fed and induce the types of organismal metabolism that your body needs to regulate glucose levels. And so what do we know about other transporters other than glucose? Well, surprisingly very little. So of the 456 transporters that I told you about, if we just survey them and ask what is transported by that transporter, almost half of them, we have very little evidence for what it transports. About a quarter of them, we have absolutely no idea whatsoever what it transports. And if we look on the opposite end, where we consider gold standard proof, where we have a structure of the protein bound to its substrate and cellular and biochemical evidence of what it does, that's only 5% of this whole family of transporters. And I told you, this is the biggest and most important family of metabolite transporters in humans. And the fact that we know so little is even more striking when you take into account that over 50% of SLC transporters are associated with human disease. And so when I started my postdoc and some of the work that I'm doing in my laboratory now is, can we systematically identify the physiological human disease? So we took this GWAS-based approach. So GWAS are genome-wide association studies. So they are studies in which you look at the different SNPs or variants in the genome of people, and then you can associate that with phenotypes or clinical outcomes that those patients have. And so we took this data set for which they had both the genetic information for 6,000 people, but they also measured serum metabolite levels for these patients. And we thought we could use this as a discovery platform to try to identify associations genetically and statistically between metabolites in circulating serum and potential variants in those potential transporters in people, and kind of use human biology as a discovery platform to kind of deorphanize these transporters. And so normally when people do GWAS, they're looking for genetic variations that are associated with human disease. And what we're trying to do here is the opposite, where we're actually looking for associations in the circulating levels of metabolites of these patients with variants in potential transporters to try to deorphanize who is transporting what in an unbiased way. And so the idea of looking at differences in serum metabolites to inform physiology is not new. So there are a number of metabolites for which there's human diseases caused by elevated levels of metabolites. So this isn't a foreign concept, but it was a unique approach that we took to deorphanize these transporters. And we first were able to show that things that you would expect from the literature that you could pick up. So for example, cholesterol is a very important metabolite, and the transporter that takes up cholesterol is LDLR, and we were able to find that variants in this transporter are associated with changes in serum levels of that metabolite. And so we next turned our attention to what was unknown, what's previously unreported. And the one transporter that I'm going to tell you about that I've been working on is this transporter called FLVCR1. It's expressed on the plasma membrane of cells. And we found that it was associated with this metabolite called phosphocholine. And we were interested in this association because it was not only associated with phosphocholine, but also the downstream metabolite of phosphocholine, phosphatidylcholine, which led us to believe that maybe